Okay, I want to welcome everybody. I'm here with David Fisher and a few others from the panel. And I'm happy to introduce Dr. Fisher, who is the professor of dermatology and pediatrics at Harvard Medical School and chief of dermatology and director of the melanoma program at Massachusetts General Hospital. David received an undergraduate degree in biology and chemistry from Swarthmore College and is an accomplished concert cellist who also received a degree from the Curtis Institute of Music in Philadelphia. David undertook PhD training with Gunther Blobel at the Rockefeller University and completed an MD at Cornell Medical College, as well as postdoctoral training with Phil Sharp at MIT. David carried out clinical training in medical and pediatric oncology at Boston Children's Hospital and the Dana-Farber Cancer Institute, where he joined the faculty. And in 2008, he moved to undertake his present position at the Massachusetts General Hospital. David identified the melanocyte-specific transcription factor, MITF, which is required for the formation and maintenance of melanocytes and is used as a marker worldwide for melanoma diagnosis. He went on to elucidate the many steps through which UV light induces melanin via a keratinocyte to melanocyte signaling network. And he defined the nature of the tanning block in red-haired and fair-skinned individuals, which stem from defects in the melanocortin-1 receptor on melanocytes. And he then identified parallel signaling mechanisms through which pigmentation in such individuals could even be restored. And further linking the UV pigment pathway to endorphin homeostasis, he defined the existence of addictive sun UV seeking behavior for those of you who are addicted to your tanning beds. And David's work on UV signaling, pigmentation, and skin cancer also spawned the translation of his many scientific discoveries to the public health policy arena, where he has been an advisor for many agencies. So with that, I'd like to introduce David and turn it over to him for his seminar. Thank you for coming, David.
Thanks very much, Chuck, and it's a pleasure to see a number of old friends and to share with you some of the work that we've been engaged in recent years. I do have a disclosure. I'm a co-founder of a small biotech company that is attempting to develop topical inhibitors of the SYK pathway. I'll actually very briefly touch on them later, with the idea that this could potentially help to darken skin and potentially protect against skin cancer as a novel strategy. So I'm going to share with you a series of vignettes, and the first of these vignettes involves a pediatric condition, the most pediatric condition I'm going to talk about today, which of course I have to emphasize when I'm with a St. Jude audience. And this is actually a fairly significant one, congenital giant nevus. This is a condition in which a benign nevus can be present at birth over a large fraction of the body. They are almost invariably associated with NRAS Q61R mutation in these cells in this region. And because these have a propensity for melanoma transformation, typically maybe a 10% lifetime risk, but these are particularly aggressive, very difficult to diagnose within the context of a heavily pigmented area. The treatment that is frequently recommended is surgery, and we're talking extremely morbid surgery if you try to remove this entire precursor lesion. And therefore, there is unfortunately a very significant clinical need to come up with alternative strategies in these children. And so we decided to model this, and we did this in a couple of different ways using mouse models. Ned Sharpless generated this allele, a lock-stop-locks allele of NRAS Q61R, and we did this then by targeting the melanocyte lineage either with constitutive Cre, tyrosinase Cre, or tamoxifen-inducible. And I'll show you where these can be valuable to us in different contexts. And you will see it's pretty unmistakable, the melanocytic proliferation that one can see. And so what we found is that in these mice, similarly, in fact, even more severely than what one sees in humans, there is a propensity after a latent period of time for melanoma transformation. And what's seen in the red part of this curve is the heterozygous state, NRAS Q61R over plus. And so after a significant lag period, by about a year, a little before a year, these mice will occasionally develop a single invasive melanoma. If we breed the NRAS allele to homozygosity, there's a much higher penetrance and a shorter latency period. So we can see similar biology eventually that occurs within these animals. So one feature, if we're going to think about trying to regress nevi, that was terribly important to us is the issue that nevi, as for many other primarily benign neoplasms, have what we believe is a proliferative phase and a senescent phase. There's an oncogene-induced senescence, complicated, very important pathway. And we simply wanted to know, firstly, does that occur here? And it does. But we wanted to know the kinetics of this because we didn't want to be treating or attempting to treat with therapeutic strategies that might be selective for one or the other of these phases, but doing it at the wrong phase. And for practical purposes, these children, we assume, and it's actually been demonstrated by others, their nevi post-birth, certainly months later, are primarily in the senescence phase. And so we analyzed using the inducible Cree, either proliferative markers, so Ki67 as a proliferative marker that's shown in the graph at the bottom in red dots, or INK4A as one of multiple senescence types of markers, and that's in the blue dots at the bottom. And what we simply found, not surprisingly, is that within the first couple of days, it's primarily proliferative after inducing the NRAS Q61R gene with tamoxifen. But then we can see a flipping, and as the lesions become increasingly senescent, they lose their proliferative expression of Ki67. And so literally within 20 days or so, less than 3 weeks, these things are, for practical purposes, essentially entirely in the senescent phase. And that was valuable for us because we wanted to be sure, if we're treating older mice, that they are remaining mostly, if not entirely, in the senescent phase. That's the condition that we think would be most clinically relevant, and not trivial, because their cell cycle dependence, for example, presumably is minimal, if at all. So we tested a whole series of agents. This is a good era to be looking at targeted kinase inhibitors. We found MEK inhibitor could actually regress these cells and challenges with delivery, but if we inject it into the skin of these mice, here you can see combination of MEK inhibitor and PI3 kinase and mTOR inhibitor combination, binimetinib and omepallisid. And if we waited months, 3, 4, 5 months after these kinds of cutaneous treatments, as you can see in some of the gross images, some of the immunofluorescence staining for melon A, a green in the panel on the left, the green fluorescence almost entirely disappears, and melanin stain at the bottom on the left two panels, you can see that the targeted therapy is almost entirely ablating that treatment as well. Some of the deeper regions were somewhat incomplete and the kinetics was rather slow, but there is evidence that these actually retain a dependency for these signaling pathways downstream of NRAS, and that is potentially a tractable strategy in these children. On the right, I'm representing some of the immunotherapy approaches that we could take, and this is actually one of multiple things that we've tested where we combined a pro-inflammatory hapten. Squaric acid dibutyl ester, SADBE, is almost like poison ivy. It's a delayed type hypersensitivity inducing topical agent. Imiquimod is a TLR agonist, and PD-1 antagonism is not unknown, I'm sure, to all of you. So we were kind of throwing the kitchen sink at these from an immune perspective, and we actually saw very dramatic regression of these lesions in the areas that were being topically treated. And this was just simple topical because they became inflamed by treatment with this hapten, and presumably the deeper inflammatory cells were recruited up towards the superficial areas where the topical hypersensitizing agent was being applied. We looked then at what components in this immunotherapy combination might actually be responsible. Apologies that there's a little corruption here in the text. And it turned out that that topical hapten had the activity and work as a single agent. So much so that if you look in the upper right images, if we treated the skin and waited 4 weeks, the left column in that right upper square shows in red melanocytes. At the bottom, in the black little wavy things, are the melanin that is present. But in the right column, panel E and F, you can see that this single agent, a month later, there are no melanocytes detectable, or virtually no melanocytes detectable, and the melanin is also gone. And so we actually use this single agent treatment to treat the entire back region, the dorsal region of these mice, and prospectively see not only could we no longer visualize the melanocytes, but would it actually prevent melanoma from happening? Are there escapers or something of that nature? And so in the tumor curve on the left, the Kaplan-Meier, what you can see is that we actually found that in little panel A embedded within panel E, you can see a mouse that it was treated is a little whiter looking, and mouse B is darker. That's the mouse that received the vehicle control. We actually had 100% protection against melanoma formation, except a few melanomas formed on these mice, but in areas that were not treated, like on the tail or on the face areas, that sort of thing. So almost a kind of positive control, suggesting that the ablation of these lesions was as complete as we could discern from this kind of prospective approach. So how would a hapten, a contact hypersensitivity inducing agent, help to regress? What lineages of inflammatory cells are required for this? And so because these are immune intact mice, we could ablate different immune populations by injecting IP large doses of monoclonal antibodies directed against CD19 B cells, CD8 T cells, CD4 T cells, NK cells, et cetera. And the only lineage whose ablation disrupted the activity of squaric acid was anti-F480, which is a macrophage-directed antibody. And this suggested that it was actually macrophages that were an innate immune mechanism of leading to the clearance of these melanocytes, these nevus cells. And when we looked further, we could see evidence of M1 polarization. This is a pro-inflammatory marker. And it suggests to us that this is indeed at least one of the essential populations of cells contributing to the demise of the nevus cells within these congenital giant nevi. So armed with the information that this agent could work without adaptive immunity, without B cells, without T cells, or dendritic cells or NK cells, but it just needed macrophages, we tried a fairly aggressive experiment, which was to take a resected human congenital giant nevus and xenotransplant these onto immunodeficient mice, the SHO strain, that is devoid of B cells, T cells, but actually is thought to have at least partial retained function of macrophages, and ask, is it possible that on this immunodeficient mouse background, the xenografts of human congenital giant nevi might respond to topical squaric acid? And what's shown on this slide is that you can see an induction of macrophages, mouse macrophages with the F480 marker within the transplanted human congenital giant nevus. And on the next slide, if we stain the treated nevus on the top is vehicle and on the bottom is after squaric acid, you can see that actually most of the nevus cells, about 90%, actually receded, regressed. And these are actual human nevi treated on the immunodeficient mouse. If you advance, you'll see the quantitation of that, and it's roughly 90%. And so we think that there are opportunities, I won't read through all the details of this, but there are opportunities to actually test this. This agent, squaric acid, is actually used in other clinical scenarios within the field of dermatology. We think there are opportunities to test this in humans, and there's actually a clinical trial under IRB review at our institution at this time. And there also may be opportunities to enhance this. We actually think that combining with targeted agents may, there's some reason to believe that may actually add to the efficacy of M1 polarization in macrophages. And so that's another area which we're looking to further investigate. So I'd like to summarize for you a perspective that we have on how how ultraviolet radiation regulates pigment production within the skin. And I'll summarize very quickly for you in this cartoon, the most important feature of which is that UV radiation, we believe, is acting directly on keratinocytes, not melanocytes. And indirectly, through DNA damage and P53 signaling, the keratinocytes then, as a downstream target of P53, express a prohormone called pro-opiomelanocortin, or POMC. POMC is a precursor of multiple small bioactive peptides, and these are then post-translationally cleaved and secreted. And one of those is alpha MSH, melanocyte-stimulating hormone. It is secreted by the keratinocyte and binds its receptor, the melanocortin-1 receptor, which turns on cyclic AMP, and cyclic AMP through CREB activates the expression of this melanocyte-specific transcription factor, MITF, which has this master regulatory activity of inducing essentially the entire pigmentation pathway. And this involves the production of special organelles called melanosomes that become filled with melanin. And we'll talk a little bit more about how melanin is synthesized from tyrosine. But ultimately, the melanosomes are trafficked back out of melanocytes into keratinocytes, and you might be able to make out the keratinocytes in this cartoon. The melanosomes are not randomly distributed, but they distribute over the nucleus on the sun-exposed side of the nucleus in the keratinocytes. So virtually all of our melanin in our skin is somewhat like an umbrella protecting the nuclei of keratinocytes in the epidermis. And the melanin does not stay in the melanocytes. That's the overall pathway of how this works. The gene that has been implicated in people with red hair, freckles, inability to tan is the MC1R, that melanocortin-1 receptor. It's a G protein coupled receptor, and individuals with red hair have essentially non-functional variants. They will not respond to MSH to elevate cyclic AMP, turn on MITF, and the pigment pathway. And it was that clue that then allowed us to kind of work upstream and downstream and piece together what this entire pathway was. So with that little introduction, I'm going to move on to the next slide and tell you a vignette relating to what it is about red hair that elevates the risk of developing melanoma. So to study this, we utilized a mouse model of BRAF V600E. This is the most common human melanoma oncogene, and there are multiple animal models. This one was generated by Marcus Bosenberg and Martin McMahon, and what we did was to induce BRAF oncogene only, not BRAF in combination with a second or third oncogene. But we did it on different genetic backgrounds. And when we did it on black mice or red-haired mice having a non-functional variant of MC1R, or albino mice that are missing the first enzyme in the entire pigment pathway, the tyrosinase enzyme, what we find is that the black mice in the black curve very, very rarely will develop melanoma, but they develop nevi. This is your common garden variety nevi of the skin. But when we did this in red mice and activated the BRAF oncogene a number of years ago, we found that after a latent period, about 50% of these mice developed invasive melanomas. And it was only by variable of having red pigment rather than black pigment, we didn't UV irradiate, and that was a rather surprising finding. Because it had been assumed that people with fair skin are at elevated risk of melanoma because they don't have good shielding against UV. But these mice never experienced UV radiation. And so it led us to ask, is it the pigment or is it something else downstream of the red hair gene, the MC1R gene? And so as a G protein coupled receptor that regulates cyclic AMP, of course, there are many pathways that MC1R loss of function might regulate, pigment being only one of them. And so we could test, is pigment just a marker of this susceptibility state, or is it a mediator? Is the pheomelanin red pigment actually are involved in the carcinogenic propensity? And we could test that by adding an albino mutation to the red hair genetic background. And what we found is if we now had albino redheads that cannot make any pigment because the tyrosinase enzyme is not present, what we found is it entirely reversed the melanoma risk. So genetically redhead, low cyclic AMP melanocytes, but missing tyrosinase, making no pigment whatsoever, completely reverse the risk of developing melanoma. And what this told us, and I can tell you, I'm not going to show you the data because we published this many years ago, but it appears to be an oxidative propensity of red pigment. And this now we also know is profoundly amplified by the action of UV radiation in combination with this species of pigment. And so there is actually an intrinsic carcinogenicity of certain pigments, and this is probably related to why melanoma risk is not restricted to sun-exposed portions of the skin. That is much more the case for other types of common skin cancers. So with that, I'm going to move to a vignette that is the opposite of what a pediatric cancer hospital typically thinks about. But I will try to convince you that there is a mechanistic connection that is kind of intriguing. And I'll talk about Parkinson's disease. Parkinson's, as you know, is a neurodegenerative disease involving the substantia nigra of the brain, for which frontline therapy is L-DOPA. And it turns out there's been long known a linkage between the incidence of Parkinson's and melanoma. And the question is why? And so I'm going to describe literally a patient who presented to our clinic at Mass General Hospital. This was a man in his 70s, red-haired, had loved to sail, had a lot of sun damage, diagnosed with Parkinson's disease and started on Sinemet, on L-DOPA therapy in February of 2017. 2 months later, was diagnosed with a melanoma. And here is the curve of his melanoma diagnostic history since 2017. He literally, in the next 2.5 years, developed dozens of melanoma. And he would come back about every 6 to 8 weeks, very frequently having a new melanoma diagnosed. It actually turns out that we went back in more detail, and his Parkinson's symptoms began at least a year, maybe a year and a half, before he started this treatment. So the correlation of when the melanomas exploded may not have been as much when the Parkinson's symptoms began as when his treatment began. So what might connect these two diseases? Could it be shared genetic risk, common pathophysiology? Could it actually be treatment of Parkinson's disease with L-DOPA? And what we believe is that it might be treatment, at least in this particular scenario. So this is the pathway through which tyrosine is metabolized to make melanin. At the lower right is red pigment, and the lower left is black pigment. Tyrosine is first oxidized by the enzyme tyrosinase to L-DOPA. This is exactly the same intermediate that in dopaminergic neurons is produced by a different enzyme, tyrosine hydroxylase, but which carries out very similar chemistry. In the melanocyte, however, L-DOPA is secondarily further oxidized by the same tyrosinase enzyme to produce a quinone, which is a highly redox, unstable compound. And this is acted upon by thiols within the melanocyte, either cysteine or glutathione, covalently reducing them to produce the precursors of red pigment, pheomelanin. That's the lower right. If the thiol reserves of the melanocyte are depleted because the flux through this pathway is too great, then there's an alternative pathway through which dopaquinone is shunted to a quinone pathway on the left that leads to the formation of the brown-black pigments of eumelanin. You can see that the pathway goes to the right towards red pigment, and this is the carcinogenic side of melanin synthesis. And if we go to the left, if the thiols are depleted, the alternative fate of dopaquinone is down to the left side and the dark pigments. And it is actually the MITF transcription factor that regulates tyrosinase expression. And as tyrosinase expression goes up, the flux of this pathway goes up. And that is why black pigment gets made when the pathway is profoundly active. And you can see that this is downstream of the red hair gene. So when that pathway is strongly induced, the thiols are quickly depleted and the cell is making primarily dark pigment. And I want to just remind you that L-DOPA is interestingly an intermediate both in the melanin pathway and in the dopamine pathway in the substantia nigra. So we wondered, if we were to feed our red-haired mice L-DOPA, might that affect pigment? surely therapeutically, a Parkinson's patient is taking this for their brain function, for their motor function, neurodegenerative function. But in fact, what we found is at high doses, L-DOPA treatment in the red mice actually turned the skin black, and we could quantify the melanin and so on. And so what we did is to ask if we took our BRAF red-haired mouse melanoma model and fed them either low or high dose L-DOPA, would this affect their risk of developing melanoma? And that is exactly what we found. And so when we fed red mice L-DOPA, and again, this is the lowest dose, we could see a doubling approximately of the risk of developing melanoma. If the mice were red hair albino mice, that risk was entirely ablated. So it was dependent upon the pigment being present within the melanocytes. And it raised the question, what happens at the high dose L-DOPA treatment in mice where these mice actually are getting a higher dose of a drug that I was just saying is actually amplifying melanoma risk, and yet, if we give more of a pro-melanoma drug, on the other hand, it also caused the skin to turn black, and we know that the dark pigment is protective against melanoma. So where would the melanoma risk track? Would it track with higher doses of a pro-carcinogenic drug, L-DOPA, or would it go with the pigment where dark pigment typically is protective against melanoma? And what we found is that, in fact, high-dose L-DOPA that turned the mice black reversed the melanoma risk and suggested to us that this risk was all throughout a matter of the red pigment. This is the endogenous carcinogenic risk, and it suggests that low-dose L-DOPA can elevate melanoma risk through the effects on red pigment production and that carcinogenic activity. I mean, you'll see, again, if these were red-haired albino mice, regardless of low or high-dose L-DOPA, it didn't matter. Without an intact pigment pathway, the melanoma risk was essentially almost entirely reversed. So, and I can tell you we've also measured with low-dose L-DOPA, we can measure increased pheomelanin. Literally, the red pigment in the fur of these mice is increased. And it suggests that actually, potentially, the manipulation of this pathway of pigment might be a strategy to try and limit the risk of melanoma in the Parkinson's setting. And so how might one do this? You know, one might imagine clinically, if a patient like this takes L-DOPA and is starting to explode with many melanomas, get rid of the drug, which is unfortunately in our patient very difficult because his Parkinson's is in very poor control. But the last bullet point, are there other strategies that one might use to try and stimulate dark melanin, eumelanin synthesis? And so towards that idea, we wondered, here is that same cartoon. Here's a melanocyte where the melanocortin-1 receptor is not functional. This is the red-haired genetic background. And we wondered if we took a small molecule that could activate adenylate cyclase, forskolin, could that rescue cyclic AMP levels and turn the red-headed mice black? And as you can see here, it does. The mice turn very dark, again, once more, and we published this many years ago. This actually can protect the mice against UV challenge by many endpoints. The problem is that forskolin is a huge charged molecule that cannot penetrate into human skin. And so we have wondered whether there may be other strategies potentially capable of penetrating into human skin and similarly modulating the pigment switch from red pigments towards brown-black pigment. And I won't, in the interest of time, delve too much into the signaling pathway, but there is a kinase, a very interesting signaling kinase, salt-inducible kinase, that phosphorylates CRTC, which is a CREB-associated transcriptional co-activator. And CRTC, when phosphorylated by Syk, is sequestered in the cytoplasm by 1433. But if Syk is inhibited or deleted, CRTC is now free to go in the nucleus, co-activate CREB, and turn on MITF expression. And so Syk activity antagonizes MITF and pigmentation. Syk inhibitors theoretically could become pigmentation inducers. And this was genetically demonstrated by Horicki and colleagues about 10 years ago. And you can see the Syk2 homozygous knockout mouse on a genetically red-haired background. The third mouse on the left has genetic partial rescue, significant rescue of dark pigment on a red-haired genetic background. And so what we did was to take small molecules. This was a collaborative project with Nathaniel Gray, a colleague from Dana-Farber, who has made many SYK inhibitors. And I'll show you more in just a second. And we could see that small molecule inhibitors of this kinase could induce MIPF expression, as shown here. And if we topically place this on the back of a red-haired mice, it's pretty easy to see the area where this was applied. It very potently darkened the mouse. You can see on an albino, it does not have any effect, as you would expect, because you don't have an intact pigment pathway. And this was real pigment because it reverses. And the pink graph, as it goes down, reflects darkening of the skin. And if you simply stop the treatment after 6 days, it fades. And it fades exactly in the kinetics of keratinocyte turnover, because the melanin is doing exactly what a tan would have done. It's transferring the melanin into keratinocytes. The keratinocytes differentiate and slough off the surface of the epidermis. And so we can see this is really imitating what we would consider the physiologic signaling pathway for melanin. And so we tested a whole series of molecules on human skin. And as kinase inhibitors, they're small molecules. Nathaniel has made dozens and dozens of different ones with smaller molecular weights and more lipophilicity to try to penetrate human skin, and actually did find several molecules that we believe are capable of penetrating and activating the production of melanin. You can see a higher powered view of human skin. This entire sort of single file layer of cells, those are almost entirely keratinocytes. And you can see the kind of umbrella configuration. This is melanin that's actually been made by melanocytes, but then transferred to the keratinocyte, where it trafficked, as I mentioned before, over the nuclei on the sun-exposed, which is interesting. This is the sun-exposed side of the nuclei of keratinocytes, but I promise you in my lab, the sun barely shines at all. And so there's some very interesting polarization to the trafficking that occurs. It's actually a fascinating question. But all of this was being recapitulated by the SYK inhibitors. And actually, there is another whole pathway of skin darkening that we just recently published a couple of weeks ago, which I'm not going to talk about, but it speaks to an interesting crosstalk between mitochondrial redox signaling and melanosome differentiation as well. So I want to touch really quickly, and I'm going to go fast in the interest of time because I'm going a little bit slow for technical reasons here. But coming back to this UV tanning cartoon, I mentioned that pro-opium-melanocortin prohormone in the lower left is the precursor to multiple bioactive molecules. MSH is one of them, melanocyte-stimulating hormone. Advance, please. But another molecule, as Chuck mentioned a few moments ago, is beta-endorphin. And in fact, we suspected that stoichiometrically, as MSH is being produced, endorphin is being produced as well. And we published now a number of years ago that, in fact, this is not a trivial contribution. It ends up in the bloodstream if one has sufficient UV. And in mice, in that paper in 2014 in Cell, we could demonstrate literally opiate addiction to ultraviolet radiation. And we even had a knockout of the endorphin component of POMC and showed that these mice entirely lacked that dependency, withdrawal symptoms, analgesic effects, and so on, every opiate phenotype that we look at. And so there appears to be a biological linkage between opiate-like behaviors and the UV tanning pathway, UV-seeking behavior, we might call it. So from a dermatology perspective, this would seem like a very odd partnership. UV is the most ubiquitous carcinogen in our environment. And we're saying that we all have a genomically encoded propensity to become addicted to it. That doesn't seem like a very good design, which means there's got to be a powerful evolutionary driver for that. There's got to be a very good reason. And it's even true in furry nocturnal animals like mice and many other species. We're quite convinced. So what would that evolutionary driver be? Almost certainly vitamin D. Vitamin D perhaps is the only good thing that UV radiation does when exposed to our skin. There is a vital step. There's a carbon-carbon bond that as I understand it, no animal in the entire animal kingdom has the capacity of cleaving that bond, and it is essential for vitamin D synthesis. And therefore, without vitamin D and without appropriate sources, let's say 10,000, 50,000 years ago, pediatric rickets would be fatal. You need vitamin D to absorb calcium in the gut, and you need calcium in order to make bone. And so this is a vital dependency. Skin cancer is not a good thing, but it's post-reproductive years. We can probably survive without it. So is vitamin D an evolutionary driver for this UV addictive behavior? And could it be that UV seeking behavior was actually life-saving in evolution by guiding, let's say, Scandinavians who were living all winter long in a cave, guiding them to get out of that cave, even if it's briefly, feel better under the sun, and thereby rescue vitamin D levels and calcium metabolism. And so we wanted to test this by looking at whether vitamin D contributes in some fashion to a feedback loop. Could it be that vitamin D levels may modulate UV signaling and UV seeking behaviors? And so we tested this in mice. This is an assay. This is a Psychology 101 assay called condition place preference. So we have a dark box and a white box, and mice can go between the white and the dark box, but they actually prefer the dark box because they feel safer in the dark box. In any case, we give them a day to get used to the setup. They can go left and right between these different boxes, and then we condition them with the light, with a fake UV light in the dark box, and we leave them in that. They were forced to stay in the dark box for a certain period of time with fake light. And then we actually shine UV radiation in the other box and force them to stay in that box for a period of time. This is conditioning them so if they develop a preference, if they feel better, we can now test what is their preference if we simply put them in the middle, open the doors to both boxes, don't shine any light. So we're just going to measure how much time do they spend on the two sides, in the white side or the black side. And what we find is that in mice that are vitamin D receptor knockout, UV radiation produces a huge condition place preference. So the mice will prefer to go to the side where UV had been. There is no UV on the day that this assay is carried out, but they have conditioned the color of that box with the presence of UV only when they were vitamin D deficient, not when they were wild type, not vitamin D, receptor knockout mice. Now, vitamin D deficiency is very difficult to study in the context of UV, because if we wanted to look at UV condition place preference with vitamin D deficiency, as soon as we shine UV light, we're going to correct the vitamin D deficiency. We can't uncouple it. That's why we did the vitamin D receptor knockout. So we decided to test real vitamin D deficiency instead, not with UV, but with a real opiate signal, morphine, because that's the actual pathway. We're not going to study the endorphin that is endogenous after UV, but we'll just give morphine instead. And what we found is that, as shown on the left, in wild type vitamin D, all of these are vitamin D receptor knockout on the left. In a dose-dependent way, the more morphine you give, the more mice love to spend time in the color box where that morphine was given. If they were vitamin D receptor knockout, as we give an increased dose of morphine, at even the lowest dose, they already spend as much time as they can in the box that is paired to morphine. So vitamin D receptor deficiency does exacerbate the morphine response, and they love it. But vitamin D deficient mice, so they've just been starved of vitamin D for a while, they behave just like they were vitamin D receptor knockout. And they too have a shift to the left, and low doses of morphine already hit the plateau where they spend as much time in that box where they learn the morphine conditioned behavior, which is actually pretty significant because it suggests that an opiate addict who is vitamin D deficient is perceiving a stronger opiate signal. And in the mice, this was reversible. If we gave vitamin D back, in the last series of curves on the right, we see that rescuing vitamin D levels in vitamin D deficient mice restored the curve, the dose response, so that lower doses of morphine no longer elicited as profound a condition place preference response. And so these data suggest that and we also had human data. We think that these data, which are also validated by some human observations, suggest that, in fact, sunlight and UV might have actually had a beneficial evolutionary benefit for opiate addiction. This could have been a place in evolution where opiate addiction was life-saving. It also could suggest that vitamin D repletion may diminish opioid addiction in human populations, and we do see that in the mice. And and lastly, could it be that vitamin D may modulate non-opiate addictions? Is this selective for opiates, or do we think it might even involve addictive behaviors more generally? And we have a little bit of evidence that it may be more general. So I'm going to go kind of quickly through this. This is a story involving MITF, not for its pigmentation effect, but as an oncogene. And many years ago, Levi Garraway, and we were part of this study, identified the MITF gene as an amplified oncogene in a small fraction, 5 or 10% of melanomas. And subsequently, we found that there was, in collaboration with several other labs, a point mutation in MITF that actually enhanced its transcriptional function. And this was also associated actually with both familial and sporadic melanoma. And it led us to wonder, are there other mechanisms that may dysregulate MITF in melanoma? And that's what I wanted to share with you. And it involves the gene MTAP, methylthioadenosine phosphorylase, which is an interesting gene located very close to the CDKN2A gene on chromosome 9p21. These genes are so close to each other, they're frequently co-deleted or co-transcriptionally silenced. But there are occasions where MTAP deletion may occur without CDKN2A, suggesting, and others have suggested for some time, that perhaps MTAP has its own tumor suppressor activity. And we can see, as if you look at the bottom, its loss in melanoma from TCGA is not trivial. It's maybe 20% of melanomas. Both copies are deleted, and one copy is deleted in more than half of melanomas. It is not restricted to melanoma, but as you'll hear in the next few minutes, we think there's something unique to melanoma that MTAP may be involved in. And so the activity methylthioadenosine phosphorylase catalyzes, it acts on a substrate, MTA, methylthioadenosine, to separate adenine and the methyl group. And we wondered whether the loss of this could be modeled in melanoma cells. And what we saw is that if we knock down MTAP, melanoma cells or melanocytes became highly, highly pigmented, as you can see from these pellets. And in fact, even if we just incubated the melanoma cells with the substrate that would presumably accumulate in the absence of this enzyme on the upper right, the pellet also became very dark. And the melanocytes, primary melanocytes, became highly dendritic. So they weren't just making melanin, but they were actually differentiating in a way where the dendricity is manifesting that. And even topically, we could put MTA on the ear of mice and we could see that it induced pigment. And so we wondered, does it have effects on proliferation? Those were pigment, but what about melanoma? And sure enough, we could see that if we knock down MTAP in two different melanomas here, where MTAP was wild type, we could see enhancement of colony growth, enhancement of proliferation. And if we took a melanoma, the GRM line is MTAP null on the upper right, and we added back MTAP, this was actually growth suppressive. And so when we see a combination of oncogenic activity and pigmentary activity, this made us wonder, this sounds like MITF. This is the kind of dual activity that MITF participates in. Next slide. And so we looked at what happens. If you look at the bottom, you can see Western blots. If we knock down MTAP, sure enough, MITF levels are increased in a bunch of melanoma lines and in primary melanocytes. And actually, Krebs phosphorylation was also increased. And I would remind you in the little schema in the upper left, MC1R upregulates cyclic AMP, which activates PKA, which phosphorylates Krebs and activates MITF. And we can see in melanomas that in fact MTAP and MITF expression in a large subset of melanomas was reciprocal with one another. Could it be that low MTAP was somehow upregulating and dysregulating the expression of MITF? And this actually turns out to be the case through a cool mechanism, we think, which is MTA, the structure, the substrate that accumulates in the absence of MTAP. Its structure is on the upper left. And cyclic AMP is on the lower left. You can see how similar they are. I mean, I don't know exactly what that means in chemical terms, but in fact, what we believe, next slide, is that MTA accumulation results in an endogenous phosphodiesterase inhibitor activity. And we can see this manifest on the right because if we incubated with increasing doses of MTA or increased time of MTA, we could see MITF induction, which was dependent on the activity of protein kinase A. And in addition, we could actually measure that this effect was associated with elevations in cyclic AMP levels. In other words, cyclic AMP metabolism was no longer happening as efficiently. We were not affecting, to our knowledge, the expression or the synthesis of cyclic AMP, but rather its metabolism. And so these are necessary and sufficient experiments, which I'm going to march through because I'm running late, simply to say that the induction of MITF by loss of MTAP appear to both be necessary and sufficient to explain the proliferative effects of MTAP loss on melanoma. And so we think this is an interesting additional mechanism to dysregulate MITF, and we have actually found some evidence, early evidence, that MTAP loss may actually have biomarker utility in various contexts of aggressive behavior or treatment refractoriness in melanoma. So my last couple of minutes, I just very quickly want to touch on the very important question of what makes melanoma immunogenic. And so, as you know, the melanoma field has experienced a revolution of immunotherapy, which has great efficacy with immune checkpoint inhibitors, largely thought to be due to UV-associated neoantigens. We tested this in a forward genetic model in which we took BRAF P10 E57 black 6 mouse melanomas and in vitro UV irradiated them and single cell cloned them and exome sequenced them and chose lines that were isogenic, which we call parental 0 UV mutations, and then several clones that actually had UV mutational burdens very similar to human cutaneous melanomas. And when shown here, grown in immunodeficient mice, they have very similar growth kinetics. But when grown in immune intact mice and then treated with anti-PD-1 checkpoint inhibitor, we see that the UV mutated clones have responses and cures, whereas the parental melanoma does not. Not a surprising result, but kind of matching, as we think, the presence of these UV neoantigens as targets for the immune response. So what we find, interestingly, is that so the parental melanoma, in a sense, may be a model for one of the key mechanisms of resistance across cancer, which is insufficient antigens. Melanoma is unique because of that UV burden that we've subjected our skin to throughout our lives. So we decided to do an interesting experiment because we had all these UV mutated melanoma mice that were cured. We challenged them with either the same melanoma, which they all rejected, or the parental melanoma that does not have any UV mutations. And what we found is that 75% of these mice could reject the parental melanoma after they had been cured of a UV mutation-bearing melanoma. And this was pretty surprising because this suggested that there has been an epitope spreading and the immune response is not restricted to the UV mutations. And so we actually tested this by, and the panel E on the left is demonstrating this. About 75% of these mice could reject and survive a challenge of the parental melanoma. And we could look in the tumor-infiltrating lymphocytes of these UV mutated melanomas and stain for T cells capable of recognizing one of the known melanocytic antigens, GP100, for which there is a tetramer that will bind and stain. And we could see that they were strongly induced within the tumor microenvironment of a UV mutated melanoma. And we could even take a pancreatic cancer that was wildly aggressive, and if we forcibly overexpressed 1 or 5 melanocytic proteins, melanosome pigment-related proteins, those two would allow the UV melanoma survivors to delay the engraftment of a pancreatic cancer if it expressed melanocytic proteins, but not if it did not have those melanocytic proteins. So we knew that it was this epitope spreading to wild-type melanocytic proteins that was occurring in mice that had mounted a successful immune response against UV-mutated melanomas. So, and I want to remind, in the clinical world, when melanoma patients receive immune checkpoint inhibitors, it is common, maybe a quarter of these patients will develop autoimmune vitiligo, which is depigmentation of what is otherwise distant healthy skin. And we suspect that this represents exactly the same type of epitope spreading phenomenon. And so lastly, we have, well, so this is the model that we have neoantigens within a tumor that is inflamed. The neoantigens are targets for T cell killing, and the T cells are present within an inflamed tumor microenvironment, but the tumor fights back because it expresses PD-L1. And therefore, you have an inflamed tumor microenvironment, but there are chemokines and cytokines as well. And because of PD-L1 expression, the T cells are not functional, but autoreactive T cells may be recruited because the chemokines are present and the antigens are present on those melanoma cells. But still, both the neoantigen targeting and the autoreactive T cells are inactive. And then when checkpoint inhibitor is applied, both neoantigen targeted T cells and autoreactive wild type targeted T cells will become liberated, capable of filling the tumor and causing autoimmune vitiligo. So finally, we have come up with a strategy to ask, can we imitate this event not by adding UV neoantigens, but by creating inflammation in that parental melanoma that does not have UV mutations? And we did this, as shown on this picture of a mouse, by injecting the tumor on both sides of the mouse, on the back, but then inducing inflammation with topical imiquimod and a fractional laser. This is a laser that pokes tiny holes and is treating about 5% of the tumor volume. It is not ablating the tumor. It is purposely sparing most of the tumor to create inflammation. And then we give anti-PD-1 and measure bilateral tumor responses. Do we get epitope spreading, and will the other tumor on the opposite side respond? And what we see in the curve with the red arrow is that this triple therapy, pro-inflammatory therapy, has greater than 50% bilateral responses. And it is dependent on CD8 positive T cells if we ablate them. And this will actually work in other cancers as well. And here we show this with a pancreatic cancer model. It has very similar efficacy. And so we think that there are strategies to overcome insufficient neoantigens through the purposeful induction of inflammation, which sort of brings me full circle back to that idea with the congenital giant nevi, where here we were using a poison ivy-like hypersensitivity inducing agent as a means of, in that case, not adaptive immunity, although we're testing that in more detail now, trying to evoke an innate immune response against the congenital giant nevi. So with that, I will finish. My next slide is the acknowledgments. There are numerous people I want to thank. On the right half are the many collaborators who have helped us with all aspects of this work. I've tried to put the names of my students and postdocs and alumni where possible on the left side, and I apologize that I've gone over a little bit, but I'm happy to take questions and also to stay longer if there would be interest. Thanks very much.
Thank you, David, for a cosmic presentation. I'm going to encourage people to enter questions into the chat. And since we're approaching the top of the hour, I'd like to invite you to stay on, David, for a continued discussion and an extended Q&A. And before we close, this 1 hour presentation and move on to the extended question and answer period, I just want to advertise that Matt Vanderheiden, MIT, will be speaking next on the influence of metabolism on cancer progression as a part of this series. So if people would enter their messages, their questions into the chat, we can just continue on for as long as David is willing to stay with us. Okay. Perhaps as the host, I'll ask a few questions, having had the pleasure of reading some of your papers and being familiar with some of your previous work. MITF was identified in micro-ophthalmic mice, suggesting that there are mechanisms that control not only the size of eyes, but my understanding is that those mice are also deaf. Is that correct?
That is right.
So how can you account for microphthalmia and deafness based on MITF activity?
Yeah, so there's some information about this. So it turns out that in the inner ear, we all have a small population of neural crest-derived melanocytes, which are required for proper hearing, and they likely are involved. There's been some research, not enormous amounts, suggesting that they maintain a potassium gradient in the inner ear, and exactly where the melanocytes came from, in evolution to be playing such a funny role in the inner ear, I have absolutely no idea. In the eye, there is also quite a bit of information, actually more information in the eye. So, as you know, there are pigmented cell types in the eye, and the retinal pigment epithelium, the RPE, which is the outermost layer of the retina, is a darkly pigmented cell type, but it is not a neural crest-derived, it's a neuroectodermal lineage. So it's a different lineage. It expresses MITF, albeit it's actually a different isoform from a different promoter of MITF. And it turns out that when MITF is deficient in that lineage, the lineage of RPE experiences very severe deficiency. I think MITF is playing a role in defining this as a pigment cell type, and consequently, when the RPE is missing MITF, it fails to fuse properly during eye development, and the vitreous, as I understand it, literally leaks out, and the eyes are literally small. So the pressure, the positive pressure that grows the globe of the eye is lost, and there is literally a small microphthalmic phenotype that occurs. So who would have thunk? There's also roles of MITF. MITF is part of a transcription factor family that involves TFEB and TFE3. And those are very interesting transcription factors involved in autophagy and in lysosome biogenesis. And they are somewhat distantly, but not that distantly related to the MYC family. They're BHLH-ZIP transcription factors as well. So these all have their roles in cancer. A number of them are also implicated as oncogenes.
Okay, so I have a question from Rakesh Sharma. He wants to know how melanin and P53 interact to induce tumorigenesis.
So melanin melanin and P53 in tumorigenesis, there's not, so P53 in melanoma genesis is actually a complicated topic. Melanoma is one of the relatively few cancers which is refractory to cytotoxic chemotherapy or ionizing radiation, but which tends not to lose P53. And it's surprisingly uncommon for melanoma. It certainly occurs, in a sizable minority, but it's not like many of the other epithelial types of cancers where P53 loss is essentially, pathognomonic of the tumor. It's not that way, despite the fact that melanomas behave in a highly apoptosis-resistant fashion to your typical genotoxic drugs. I've always interpreted this, and this is somewhat of my speculation, that melanocytes are survivors of UV. And I suspect that they have evolved a capacity to withstand a barrage of DNA damage throughout their lives and have a different set point in the apoptosis threshold. and there are people who have looked at things like BH3 only assays. Tony Latai, for example, has these very interesting mitochondrial assays that have looked at how close is the cell to the threshold of going over into apoptosis. And I think melanoma cells are far. I think they've set their apoptosis threshold at a different place, so that I'm not sure their dependency on p53 is quite the same as for many other types of cells. I think on the melanin side, as I mentioned before, I think pheomelanin, red pigment, has a carcinogenic propensity through this redox mechanism. But I don't think, normally if I'm giving a talk in person and I look out in the audience and I see a couple of people with red hair, I'm very quick to add, this is not a very high risk of melanoma, because these are rare events. It's measurable, it's statistically significant, but if it were common, the incidence of melanoma in red-haired human beings is still really low. I mean, think of how many melanocytes you have. Think of how, even, the incidence being, that is the pigment variant in humans, the normal variant of pigmentation that has the highest risk of melanoma, but it is still relatively low. Hopefully most red-haired people won't get a melanoma, and if they do, it will be one, maybe two, it will be caught early and they'll be cured. So I think it is a carcinogenic risk, but what I think is also interesting is, what does this tell us about protecting ourselves from the cumulatively higher risk, like UV radiation, sun exposure, things of that nature, which are preventable. And I think there, we could talk about sunscreen if anybody is interested. There are some big challenges right now in this entire industry, where it was discovered that sunscreens are making their way into our bloodstream, even when used properly. FDA is in turmoil, the industry is in turmoil. Sun protection is terribly important, but the best way to do that is not entirely clear.
Okay. Martine has a question about p53 loss. Wondering what happens in response to UV irradiation in the absence of p53. I mean, to what extent does p53's control of POMC matter, or are there parallel pathways that bypass p53?
Yeah, so in the absence of p53, no tanning. So that was actually in one of our early studies 15 years ago or so. So we used those mice for that. We've looked at keratinocyte conditional p53 knockouts, and in fact, not only can you block the tanning response, but you can block the addictive response as well. And we showed some of this back in, I think, in one of our other papers. So the keratinocyte p53 response is unique, and actually we think that there is a lineage specificity to that p53 regulation of POMC. We haven't looked at numerous lineages. We looked at, I think, fibroblasts and melanocytes because they were convenient, and we did not see much of a POMC induction in the absence of p53. In keratinocytes, it was like 30 or 40-fold induction. It was like huge, massive induction. And it also comes way down to baseline in the absence of UV versus with UV. It's anything that when someone tells you there's a 40-fold induction, you have to wonder, what did it start out as? Was it, if you go from 0 to barely detectable, that can be 100-fold. And so, part of the magnitude of induction quantified is just how low it was to start with. But I think in our skin, actually, the keratinocytes really downregulate this pathway, but then it is very robustly inducible. And interestingly, other lineages express some POMC independent of p53 at a constitutive level that is higher, but they don't respond to DNA damage, and they don't seem to respond in a p53 dependent way. So there are lineage-specific mechanisms, and I've often wondered, we don't really know much about it, but how is endorphin regulated in the brain? When people go outside and exercise, and then they get an endorphin high. I always thought it's from the sun. They're just getting a tan, and I'm kidding. Nobody believes it was from the sun. They think it's from being a good exerciser. But actually, there probably are other neural networks that are feeding into POMC expression. POMC is, of course, a very important hormone in the pituitary and part of the hypothalamic pituitary adrenal axis as well.
So David Sotillo addressed a comment to you which I think I want to read. It says, outstanding talk. Thanks providing a biological explanation to my need to watch this seminar from an outdoor sunny location. Somebody wants, Helen Fillmore wants you to comment on differences between ocular melanoma and I guess the usual melanoma. She wants to know if the difference is due to the cell of origin. I know the mutations are different in ocular melanoma, that there's a different protein receptor involved, but can you comment further?
Yes. So fortunately, so ocular melanoma, there are many cell types in the eye that can develop melanoma. The most common uveal melanomas actually are neural crest derived, but they are not UV associated because the sun isn't making it back there efficiently and is not, these are very low UV mutation burden tumors. And that is probably related to the lower incidence of them as compared to cutaneous melanomas that are experiencing a lot more UV radiation. The density of mutations and neoantigens is probably the closest predictor, or I should say explanation, of why immunotherapy with checkpoint inhibitors is largely unsuccessful. Not entirely unsuccessful, but overall significantly less successful in ocular melanoma, and then other low UV versions of melanoma, such as mucosal melanoma along the GI tract or the GU tract. Those melanomas, again, much rarer tumors, but also fundamentally different primarily because they are less immunogenic. They have fewer neoantigens. And again, that last vignette I mentioned about artificially increasing the inflammatory microenvironment, we think is an interesting place to consider for future studies of trying to rescue epitope spreading and an immune response against the lineage, and to see if some of that might actually be beneficial in these patients. So there are other features that are very distinctive about ocular melanoma, probably beyond the purview of our talk right now, but a high propensity to spread to the liver, the very intriguing tumor suppressor pattern and involving very specific genes that are, you're absolutely correct, quite different from your typical cutaneous melanomas.
Martine asks, how close are we to have a SYK inhibitor added to sunscreens? How dark will Caucasian people's skin become?
So for this, I have that disclosure on my first slide. So in fact, Nathaniel Gray and I did start a little company. There are no products at this point. The very first clinical testing is aiming to start over the next year or so. It's all complicated by whether this would be cosmetic versus drug, prescription, et cetera. So what I can tell you is in mice, red mice could turn very dark black. I think much darker than would be cosmetically desirable necessarily, just in terms of the change of pigmentation. One thing that I think from a cancer, my interest in this really is that primarily the cancer prevention opportunity for people who have very light skin and don't respond, with a dark pigment response to UV, is that, in fact, if you look at human populations, protection against UV-associated skin cancer is present before one reaches very, very strikingly dark skin. Hispanic, Asian, Mediterranean already, where there is visual darkening compared to red-haired people or somebody like myself, but by no means highly heavily darkened skin. Already there is a very strikingly lower incidence of UV-associated skin cancers, and nowadays it used to be in human evolution that skin pigmentation was relatively matched to UV intensity of exposure. Equatorial was associated with human populations that were dark, and Scandinavian and redhead Irish and so on were, the other extreme. Now, for practical purposes, we're in the jet set, everybody moves everywhere. And so there are populations where this is all normalized. So I think the how dark would one be, how dark would one want to be, What would the motivations be? Even if the motivation would be just simply not to go sun tanning, would be great. I would say the one thing that is available today is to use a vitamin D supplement rather than tanning as a way to maintain a vitamin D level. There are indoor tanning beds that advertise, liquid sunshine. I don't know, how do they describe it? The tanning bed is your healthy source of, essential vitamins. And what they also do is they give you the first 10 doses for free so that you become addicted, and then they charge you for it. So there are lots of tricks in the industry, unfortunately. But I think if there were behavioral opportunities to sway people away from UV carcinogenic, vitamin D is certainly present and induced by UV radiation, but there is a non-carcinogenic way to get vitamin D as well. And it's very cheap, very inexpensive, and highly effective.
Okay, your hair is still dark and mine has turned gray. What is the defect in my hair follicles that prevents the survival of, I assume, melanocytes that contribute to coloring of hair? And since you've done experiments showing that topical application of certain pharmaceutical agents like forskolin, for example, or other SYK inhibitors act downstream of the melanocortin receptor, could these be used to turn gray hair brown or black?
So,
topically, of course, and shampoo.
We have, yeah. So we know a fair bit about the mechanism, not so much about ways of reversing it. So I'll share a little bit of background here. So we've actually studied this area a fair bit. And I think in 2006, we had a paper in Science that demonstrated in mouse models and also in human aging, so senile hair graying, and that's, of course, not pejorative in any way, that what is going on in the hair follicle is hair follicles go through cycles. It's a cyclic process. The follicle grows, and then the factory portion that makes the hair and the pigment actually dies. It undergoes a regressive phase, a resting phase, and then there are stem cells that regenerate that follicle. And so there's a constant regeneration. And the reason our hair turns gray with time is that the melanocyte stem cells are not perfect stem cells. They're imperfect. And the number we could see was diminishing over time. And my postdoc at the time, Emi Nishimura, went on and started her own lab and did beautiful work and identified a number of environmental insults that will accelerate the loss of those melanocyte stem cells. And then just recently, Yaxie Xu at Harvard, and we collaborated with her in this study, identified another environmental context in which there is a dramatic acceleration or a slow acceleration, depending on the insult, of the loss of the melanocyte stem cells, and that is stress. And what she did, this was published in Nature a year and a half ago, I think, or so, either 1.5 or 2.5 years ago, in January of 20, maybe 19, something like that. And what was shown is that by literally stressing mice with a whole bunch of different stresses, it could be isolation, it could be water, it could be a painful injection, a whole bunch of different things, through sympathetic innervation, was activating beta-adrenergic receptor signaling on the melanocyte stem cell. And beta-adrenergic receptor, through noradrenaline signaling, is exactly the same family of that as that red-haired MC1R gene, that receptor. And in the stem cell, by definition, it is undifferentiated. Very low cyclic AMP, very low MITF. It's not making pigment. And the model that we have, and we still have, is that the shock of a sympathetic outburst results in a burst of cyclic AMP, inappropriate MITF upregulation that epigenetically takes that stem cell out of a stem cell state permanently. And we are endowed at birth with a finite number of these cells. In the mouse, it's like, depending on which hair, if it's a whisker or dorsal or whatever, they're like 10 to 30 or something like that. And in human, again, it's a limited number. And so the model is that as you are stressed through life, and it can be a stomach ache, it can be a grant that you're writing, it could be, your audio didn't work on your talk for St. Jude's, whatever it is, boom, there went one of my stem cells, there went another of them. So that's the bad news, is that it's loss of stem cells, and when your hair is white, you've lost the cell. So you can try to rescue the pigment pathway. If there are no melanocytes there, it's not going to be available. The problem now, as I think about it, is actually more akin to vitiligo. How do you deal with unpigmented skin, or in this case, hair follicles? Could you get melanocytes back? And I won't share with you exactly where we're going, but there's some very interesting, the fact that you have a stem cell pool is pretty cool. You have a stem cell pool, and what I'll just share with you is that in autoimmune vitiligo, repigmentation can occur in these patients if you immunosuppress in the active phase. But the way repigmentation occurs is really interesting. The way it works is if there are hair follicles embedded within that epidermal skin that has lost its melanocytes, when you immunosuppress, melanocytes come out of the hair follicle, each hair follicle, and in a perifollicular fashion, start to crawl out, repigment the skin, and if you're really lucky, they will coalesce and fully repigment. So there is actually a reservoir of melanocyte stem cells that retains the ability not only to make hair pigment, but even epidermal pigment. So could one trick those cells to migrate down to the adjacent white hair follicle? Who knows? But these are all interesting questions.
So just following up on this point, what happens to hypertensive patients who are chronically taking beta blockers? Is there any effect on hair pigmentation at all?
It's an extremely good question, and we wanted to ask that. The problem in a clinical scenario is it's not going to reverse the loss of melanocyte stem cells. If they're gone, they're gone. But what it could do is change the kinetics of hair loss, not now, but 30 years from now, when you're down to your last stem cell per follicle. It's just not an experiment I think I can study, but it's a great question.
Very nice. I keep looking at the chat box, and I don't have other questions coming in, but I have so many. So if you don't mind, I'll just keep you for a few more minutes.
Sure.
Just for my personal amusement, I'm fascinated by the MTAP story because as a maven, somebody who studied from the very beginning CDKN2A deletions, I know there are small deletions and large deletions, and many of the deletions, even the small ones, include MTAP, which sits inside of the TAD insulators in that locus. And this was the first time I've ever heard an explanation for why MTAP might be a tumor suppressor and why MTA, acting as a phosphodiesterase inhibitor, if I have it right, could increase cyclic AMP-dependent proliferation. Did I get it correct?
Yes.
Are these data published?
We're about to submit it.
I see.
That's exactly right. So no, it's not published.
I think this is a very important story because those genes sit on top of each other, and there's been some additional discussion that large deletions, which go across the TAD boundary, also include interferon genes, which are in a different transcriptional unit. So I wonder if some of those data really are perhaps being overinterpreted by the fact that all of these deletions, large deletions, all include MTAP, and many of the smaller ones include MTAP. And so this is the first I've heard of trying to associate MTAP with deletions. MTAP deletions must be common in somatic melanoma, therefore, right? The ones that are deleted are mutated.
Yeah, I mean, if you look at the expression level, it's super common. It's like RNA levels, are down in 70% or something like that. So, On the other hand, I'm not sure what the role of cyclic AMP is in proliferation for many cancer types.
Yes.
We were particularly focused on MITF because that is a direct target of CRIB. And MITF is such a well-validated oncogene by point mutation, by amplification, and so on. And that, from all of our work has really looked at this in the melanoma context. MITF is actually associated occasionally with other cancers, but not commonly. So I'm not sure if MTAP loss in other cancers, just maybe is a synthetic lethal opportunity for people with, looking at other methylation pathways to try and drug. I know that has been another area to look at. So maybe this is unique to melanoma, maybe it's opportunistic, or maybe it will be shared by other cyclic. I mean, there certainly are other cyclic anti-oncogenic pathways.
There are MTAP knockout mice, but I don't know if anybody's looked at MITF in them. In fact, I don't even know who's made them. I just know they're out there, and that there are a few papers claiming that MTAP alone is a tumor suppressor. I don't know how good those data are, but it would be interesting to know whether the MTAP deletes, pure MTAP deletes, INC4A positive, have MITF defects. I think that would be interesting.
Yeah.
Okay, I have another question from Artie Mustaki. Do you have any insights on the secretome of senescent melanocytes? Senescent cells are considered inflammatory. What do you think is blocking the activation of macrophages in the absence of hapten treatment? Or do you think the melanocytes are resistant to macrophage cytotoxicity, let's say by increased autophagy of melanocytes? That's a real zinger.
It is a fantastic question. I think that is a fascinating question and something that we have thought about a great deal. The autophagy arm is thought, there are very strong suggestions that this resides downstream of MITF as well. And absolutely, the senescent associated inflammatory microenvironment is very plausibly present. We do see that it's easy to recruit a profoundly greater macrophage presence that is associated with the regressions that we saw. But I've always wondered why, what is it about these lesions? Why, even in the context of vitiligo in melanoma patients receiving checkpoint blockade, it turns out, and this may be relevant to your question, it's not just any old autoimmune vitiligo that occurs in those patients. Typically, when they develop the patch of vitiligo, it is more likely to occur in previously sun-damaged skin. So it's typically in places with prior UV damage, which may mean that there are tissue-resident populations and perhaps macrophages and other cells that have already populated that area. So I don't have any solid answers to your question, but I really love the question. I think it's a fascinating one. And I also think it's interesting, not necessarily only for nevi, but for other benign senescent neoplasms that perhaps could have similar microenvironmental conditions and opportunities, for impact. But a great question, I think.
I just have one last one. Carol is warning me that we have only 2 minutes left. You haven't played around with CSF1 receptor inhibitors, have you? Like to prevent macrophage activity?
Right. We did F480 ablation, but we didn't use CSF1 inhibitors.
Okay. So Carol wants to thank you, and we all want to thank you for staying for the extra half hour for the Q&A. It's been really a very exciting talk. I want to thank you personally for accepting Martine's and my invitation to speak. I think it was just an amazing seminar. And so thanks for your time. I know how busy you are. We really appreciate having you. Applause.
Well, my total pleasure. Great seeing you, and my very best to everybody. Stay safe.
Thanks
so much.
Thanks again. Okay.
Take care.
Bye.
Thank you. Bye.